Donc bonjour, merci de me permettre ce petit webinaire et cette introduction à RNAlead et à la technologie FlashRNA, donc, qui a été développée par la compagnie. Okay, so I'm going to start with a brief introduction on the RNA delivery problematics to help you understand the challenges of RNA delivery for next generation therapies. If you look at this representation of all the applications that now can use mRNA therapeutics, you see that there's not only vaccines that were used for the COVID pandemic, but also cell and gene therapy, regenerative medicine, immunotherapeutics. But all those treatments require different modalities. We need repeat dosing, we need high dosing when and a control immune response when the vaccines need to activate the immune response, and we need to target a variety of tissues. So all these explain why the delivery systems now need to be adapted, and the market is still in need of some alternatives. So those specific needs to expand the RNA-based therapeutics beyond vaccines are an increase of the cell transfer efficiency. We need to improve the stability of the mRNA as well, and the magnitude and the direction of the protein expression coming from these mRNAs, and all that with a controlled immune response. And that's not that easy. Currently, there is different vectors used for gene delivery. They function differently on the target cells. You have the viral vectors. They penetrate the cells via receptor interaction, and then they deliver the genetic material using either endocytosis or fusion mechanisms, like lentiviral vectors. The non-viral vectors use endocytosis mechanism to enter the cells, and even if they are known to improve the safety and the loading capacity of the viral vectors, they still face a major limitation, which is the endosomal escape, as only 2 to 3% of the RNA carried by the LNPs, for example, finally get to be translated into proteins. So at RNAlead, we have developed an innovative RNA delivery technology, which is called FlashRNA. FlashRNA are particles derived from lentiviral vectors. They can carry multiple RNAs, and they enter the cells through an efficient receptor-mediated interaction, leading to a high level of protein translation. So what is FlashRNA? It's an hybrid nanoparticle because it's derived from the combination of two natural systems, the lentiviral vectors, which bring their high transfer efficiency, a low immunogenicity, and we have modified them to get them to be completely non-integrative. And we have increased the carrier capacity thanks to the use of a bacteriophage encapsulation system, which allow to carry multiple and different RNAs into a single particle. So FlashRNA technology is a disruptive alternative to the non-viral RNA transfer solutions. It's proven now from more than 10 years of in vitro and in vivo validation. It's proprietary. And finally, it's clinic ready, as the scaling up of the GMP grade production has already been done, and we are now ready for a first-in-human trial very soon. So what makes FlashRNA different? One important thing to remember is that FlashRNA is a biological delivery technology. So it's biologically manufactured by a cell process. So our RNA are synthesized by producer cells, and they are biologically modified, avoiding all intracellular cell immune responses against them. The bacteriophage encapsulating system, which is used lead to an active packaging of the RNA of interest. And we are able to carry high payload RNAs, at least of 10 kb size. We kept the capsid coming from the lentiviral vectors, so the RNA are protected. And this ensures that most of the RNA will reach the target cell's machinery to allow an efficient protein synthesis. The manufacturers are human producer cells, so the particles membrane comes from those cells, which ensures low immunogenicity of the particles. FlashRNA particles are surrounded by an envelope protein, which allow not only a receptor-mediated cell entry, but also a fusion mechanism with the endosomes membranes, which ensures a very efficient RNA release into the cells. So we don't have the problem of endosomal escape. And finally, this enveloped protein can be engineered in order to modify the tropism of the particles and to get to some cell specificity. And finally, as I mentioned, we know how to produce the FlashRNA particle at GMP grade for drug product use in humans. So this slide just illustrates really briefly all the in vitro studies that have been performed using the particles on cell lines, primary cells, rodent and human cells. Same kind of illustration for the in vivo applications with more than 20 studies performed, mostly in rodents, and 10 administration routes evaluated so far. A few words on the first in-human trial. So in this trial, the FlashRNA particles were produced as a drug product to be delivered intradermally in patients suffering from secondary lymphedema, which is a condition affecting up to 25% of breast cancer patients treated by surgery. There's no cure currently. And in this case, two different mRNAs will be carried by the FlashRNA, by the product, in order to restore the lymphatic pathway and to treat the fibrosis at the same time in these patients. Just a quick look at a few translational insights. FlashRNA positions itself as a disruptive alternative to gene delivery platform, and its versatility allows it to be used for many different applications, from gene therapy to vaccination, to gene edition, cell reprogramming, or immunotherapy. A few examples very quickly. So this is a use of the particle expressing a reporter for subretinal injections into mice eyes. Here, we used it to engineer induced pluripotent stem cells, showing a very efficient reporter expression in all the cells with no toxicity, and a transient protein expression, which is directly related to the half-life of the protein. For gene editing, when you use FlashRNA, you have an all-in-one efficient and safe solution in hand. The guides and the nuclease are carried together in a single particle. Large size nucleases can be efficiently loaded into the particles, and as it's an RNA delivery system, there is only a transient expression of the nuclease, ensuring very low risk of off-target editing or genome damages. So those are two references illustrating the efficiency of the technology on iPS cells editing and in vivo transfer of a base editing system in a mouse model of progeria. Here, this is an illustration of the absence of inflammatory response after intra-articular or IV injections into animals. So all the level are below the threshold of detection. And finally, those are some data showing antigen delivery First, in a model for tumoral development, so we show that we control the development of the tumor after injections of the particle expressing tumoral antigens specific of these tumors. And this is an example of a vaccination with antibody secretion responses followed up after 3 rounds of injections. So as a take-home message, I'd like to highlight the fact that FlashRNA is designed to reach a balance between efficacy, safety, and scalability, which is really balanced in the technology, thanks to the association of the different properties. So the high payload, the biological RNA, and the low immunogenicity, no genome integration, of course, an active packaging of multiple RNAs and a specific packaging, high cellular entry receptor mediated, leading to the high expression level of the protein of interest, a viral envelope that can be redesigned to get some cell targeting specificity, transient expression that can be increased because we can use self-amplifying, trans-amplifying RNAs or even circular RNAs to be carried by the technology, and an easy scaling up for clinical applications. Thank you.